Hello, my name is Kelly Schuette, and in this video I will be discussing the laureates of the 2017 Nobel Prize in Physiology or Medicine. The prize was awarded to Jeffrey C. Hall, Michael Rosbash, and Michael W. Young for their discoveries of molecular mechanisms controlling the circadian rhythm. Circadian rhythm describes the regular, daily physiological changes that occur within the human body. We have known for a while that organisms on Earth have adapted their biological clocks to match the Earth's rotations, allowing for optimal hormone, temperature, and metabolism function over a 24-hour cycle. However, it was not until the discoveries of these Nobel laureates that we knew of the mechanisms that control these biological clocks. Using fruit flies as their model organism, Hall, Rosbash, and Young were able to isolate genes that control the normal daily biological rhythm. In 1988, Rosbash and Hall were able to isolate the first gene, termed period, which encodes a protein termed PER. This protein is accumulated during the day and degraded during the night via an inhibitory feedback loop. The figure on this slide shows the sequence of events during a 24-hour oscillation. Essentially, mRNA is made when the period gene is active. This mRNA travels to the cytoplasm and serves as a template for PER production. As PER protein builds up in the nucleus during the day, it blocks a period gene, thereby preventing its own synthesis. This creates a cyclic rhythm for the production of the protein. I mentioned that the PER protein builds in the nucleus, but how did it get there exactly? That was the next question on the three Nobel laureates' minds. It was answered when Michael Young discovered a second clock gene, termed timeless, that codes for a protein termed TIM. The diagram on this slide demonstrates the process of TIM in blue binding to PER in yellow, allowing for entry into the nucleus and blocking the period gene to close the inhibitory feedback loop. Michael Young also answered another key question. What was controlling the frequencies of these oscillations? The biochemical processes involved in transcription and translation are generally pretty fast, so there needs to be something delaying the process to match a 24-hour clock. Young found that another gene, termed double time, coded for a protein termed DBT, which delays the accumulation of PER in the nucleus during the night. DBT binds to and phosphorylates PER, making it unstable and degradable. The diagram to the right shows this process in fruit flies compared to a similar process in mammals. We can see how some of the PER gets degraded and some enters the nucleus, providing a delay in the accumulation of PER. This delay allows the oscillations to match the 24-hour day cycle. Circadian biology has developed into a vast and highly dynamic research field, with implications for our health and well-being. Since circadian rhythms regulate everything from sleep patterns to feeding behavior to hormone release, understanding their mechanisms has huge implications for healthcare. The seminal discoveries by the three laureates in the 80s and 90s prompted further research that uncovered the stability and function of biological clocks, as well as other proteins that influence the activation of the period gene and the way light synchronizes the clock. Expanding upon this, research has found how the suprachiasmatic nucleus in the hypothalamus of mammals uses light information to relay messages to other regions in the central nervous system. A growing body of evidence suggests that the functional integrity of the suprachiasmatic nucleus contributes to health, well-being, cognitive performance, and alertness. Its deterioration is a risk factor for neurodegenerative disease, cancer, depression, and sleep disorders. Another expansion on the Nobel laureates' research shows that circadian rhythms impact the efficiency of drug absorption due to the altered activity of enzymes. For example, most lipophilic drugs seem to be absorbed faster when the drug is taken in the morning compared with the evening. Additionally, the higher blood flow fraction an organ receives, the higher the rate constant for the transferring of drugs out of the capillaries. Since blood flow is higher in the mornings and lower at night, research in this field can help better tailor prescription instructions for patients. Thus, understanding research in this field has and will continue to increase the public understanding of circadian rhythms as it relates to our health. The figure to the right displays the key bodily changes occurring at each stage of a daily cycle. Being in tune with these changes will not only help us understand our body biologically, but allow us to make choices that align with its optimal functioning for certain tasks. Jeffrey C. Hall was born in 1945 in Brooklyn, New York. During his undergraduate research at Amherst College, he became interested in the genetic mechanisms of fruit flies. He received his doctoral degree in 1971 at the University of Washington in Seattle and carried out his postdoctoral research at the California Institute of Technology from 1971 to 1973. He joined the faculty at Brandeis University in 1974, where he noticed the fly's periodic singing pattern, which led to the discovery of the period gene. In 2002, he became associated with the University of Maine. Hall also served as an editor for several scientific journals and was an elected member of the American Academy of Arts and Sciences in 2001 and the National Academy of Sciences in 2003. Michael Rosbash was born in 1944 in Kansas City, Missouri, and grew up in Boston, Massachusetts. He received his doctoral degree in 1970 at the Massachusetts Institute of Technology. During the following 3 years, he was a postdoctoral fellow at the University of Edinburgh in Scotland. Since 1974, he has been on faculty at Brandeis University in Waltham, Massachusetts. In the 1970s, Rosbash became interested in the influence of genetics on behavior. This began his collaboration with Jeffrey C. Hall, a friend and colleague at Brandeis, where they isolated the period gene. He was an elected member of the American Academy of Arts and Sciences in 1997 and the National Academy of Sciences in 2003. Michael W. Young was born in 1949 in Miami, Florida. He received his doctoral degree at the University of Texas in Austin in 1975. Between 1975 and 1977, he was a postdoctoral fellow at Stanford University in Palo Alto. Since 1978, he has been on faculty at the Rockefeller University in New York. During the 80s at Rockefeller University, he researched the molecular basis of circadian rhythm in fruit flies. Expanding on the work of Hall and Rosbash, he showed that introducing a fragment of DNA from the period gene locus into the genome of period mutant flies restored circadian rhythm, thereby demonstrating the functional significance of the gene. He was an elected member of the National Academy of Sciences in 2007. In addition to their Nobel Prize, all three men have shared numerous other awards, including the Gruber Foundation Neuroscience Prize in 2009, the Louisa Gross Horwitz Prize in 2011, and the Canada Gairdner International Award in 2012. Thank you for listening to this presentation. Here are my works cited.